Speaker is Dr. Andrew Sutherland. So we are in the same program. Both of us are IPM advisors. He covers the Bay Area. I cover the Southern California. So Andrew has a lot of experience in structural pest control and has worked on termites, ants, and this is a very interesting project that Andrew is going to talk about on biting mites because we don't have enough data. And I'm so glad that he's doing the research. He got the grant. And thank you again, Andrew. Thank you so much for agreeing to talk. So please go ahead and get started.
Thank you, Sivash. Can you guys hear me?
Yes, perfect.
All right. Okay, so I would love to talk about biting mites. As Sivash mentioned, there's not a whole lot known about these critters, so I'll share what I've observed and recorded over the past couple of years in a sponsored project. And I will focus on prevention and monitoring. If you'd like to learn more about me and my program, you can visit my website. It's you can generally Google Urban IPM, and I'll be one of the first returns. And you can learn about the different projects that I have going right now. My work is to serve pest control professionals through research and education in order to protect communities and the environment. And I do this by teaching what I call the central tenets of Urban IPM. We can apply this process to any urban pest. It begins with education. The example here is Turkestan cockroaches, learning that they live outside rather than inside where we might see them. Prevention, and you see here the use of copper wool to prevent rodent invasion through a pipe penetration. Monitoring, simple use of glue boards or sticky traps to assess cockroach populations. Thresholds, meaning we determine when action is required. Multiple tactics, meaning we use lots of different tools, not just pesticides or not just non-chemical tactics. The example shown here is an electric heat treatment for bed bugs. We integrate these tactics and we evaluate. This feeds back into our education, and IPM never stops. We are constantly improving our IPM programs. So the story here about biting mites begins with what we call unexplained dermatitis. So your client or someone else complains of bites. I'm getting bitten. What bit me? And even my wife does this to me where she says, look at this on my skin. What bit me? And the problem is that you cannot identify a biting pest from dermatitis. You require a specimen in order to identify a biting pest and move forward. Specimen or sometimes signs, like bed bugs, we can find shed skins or fecal spots or eggs. But you must identify the pest in IPM in order to move forward with your management program. And no matter what people may tell you, there's no such thing as a typical dermatological reaction. So you can't look at bumps on someone's body and say, that's a mosquito, that's a bed bug, that's a mite. There are a lot of common culprits, or what we call usual suspects here. All right. And we can consider those, but we can also consider that dermatitis might be caused by the environment. There are environmental irritants that can cause unexplained dermatitis. And always we will consider whether we're dealing with what we call delusory parasitosis, which can be psychological in origin or environmental at first and then psychological. It could be initiated by a real infestation and then become psychological. And I think oftentimes when we have unexplained dermatitis and we cannot find one of these usual suspects, we will often consider, is this delusory parasitosis? In the case of biting mites, they are very difficult to capture and sometimes very difficult to identify. And so I will posit that there are cases that pest control professionals consider to be delusory parasitosis, when in fact there are biting mites present and we are unable to capture and identify them. I think this is a very common pest system throughout the world. And so I believe it's unreported, or underreported. What's happening with these biting mites? They are pests, parasites of their primary hosts. And so most of the time we're talking about rodents or birds that are nesting in or on or near a structure where humans spend time. They feed on the blood of these primary hosts. But when the primary hosts are removed or impacted in some way by control measures, these mites can wander and they can feed on the blood of humans as well. When that happens, they do produce significant dermatitis, especially on some individuals with sensitivity. This, of course, does have connections to delusory parasitosis or delusory infestations, as I mentioned. And you can find all kinds of misinformation online. Birdmites.org is one of these sites that has a little bit of factual information mixed in with a lot of misinformation. And I think that this misinformation is out there because there's a real need to explain these cases of unexplained dermatitis. The general ecology here is that the mites live in the nest or sometimes on the host, or sometimes they're traveling between the nest and the host, and they're feeding on blood or other body products of those primary hosts. They can disperse on the host themselves. So think about birds that fly from place to place, or sometimes even from country to country during migration. They can include these mites on their bodies that are riding, getting a free ride. But the mites can also crawl on their own. And the really scary thing here is we know from veterinary pest management research with poultry, they can wander hundreds of meters away from their primary host nest in search of blood. They will eventually starve and die without their primary host, but this takes time. So in the case of an adult fowl mite or rat mite, they can survive for 60 days or sometimes more without access to their primary host. And that whole time, they could be feeding on humans or other incidental hosts. A big question out there is whether humans can actually serve as primary hosts. And so, so far, all of the literature that's out there reports that the life cycle of these biting mites cannot be completed on humans. It requires the primary host, which will be rodents or birds. But humans are incidental hosts. There are other mammals that can serve as incidental hosts, providing blood meals to these wandering mites. Another question that we get a lot is whether or not these mites can transmit human disease. It's never been demonstrated, either in the lab or the field, but you can isolate pathogenic bacteria from these mites. If you crush them up, you can find the bacteria within their bodies. So the potential may be there, but it's never ever been demonstrated. So really what we're dealing with are incidental bites when a primary host is present in a human environment and the primary host has been removed or disturbed. So there's a few different taxa that are potentially involved. I like to divide these into indoor biters that are commonly causing problems in homes and other structures, and landscape biters, which you may pick up while gardening or hiking. And, really they can be dealt with in different ways. We have much more control most of the time over our indoor environments. And you can sustain a lot more dermatitis if you have problems with these indoor biters. So with the indoor biters, we're talking about mesostigmatid mites. There's a couple common culprits that I'll cover here in a second. And then with the landscape biters, we're generally looking at a different order of mites, the Trombitiformes. And some of these are very, I think, familiar to some of the participants here. For instance, chiggers, which are larvae of tromboculid mites, very common in many parts of the world. So let's look at the most common species here. The tropical rat mite is by far the most common culprit here in the San Francisco Bay Area. These are nest parasites of rodents, especially rats. So both roof rat as well as Norway or sewer rat, but you can also find them on domestic rodents. There was a study done in Europe looking at hamsters purchased from pet stores, and 40% of them had tropical rat mites, either on them or in the bedding after purchase. So domestic rodents can also be a really serious source of these mites. Some squirrel species are also known to carry these mites. Two of the life stages, the protonymphs and the adults, feed on blood. The other stages do not feed. And a lot of times people think that these mites are invisible, but unless you have impaired vision, you can see them, especially if they're full with blood. They get engorged and they darken in color. But generally, we're talking about 0.5 millimeter to a fully engorged one, maybe 1.5 millimeters. And so that's about the size of a period in 12-point font. If you print that out, you can see it, especially if it's moving around. These mites have incredible potential for reproduction, especially considering a reproductive strategy they use called arenatoki. And this is where a female can actually lay unfertilized eggs if she has yet to mate. Those unfertilized eggs produce males, and she can actually mate with her sons to produce fertilized eggs that will yield females. So all it takes is one female, whether or not she's mated, to initiate a population. And I've already mentioned that these guys can wander a fair distance away from their source, and they can persist for quite a long time without their primary host. Another species, or two species here that we group together, are the fowl mites, northern and tropical fowl mites. And these guys are associated with birds. We find them as a primary and significant pest within poultry production, so chickens, turkeys, and other fowl. But you can also find them on commensal and migratory birds. So think about pigeons and starlings and swallows, and really over 70 species of birds have been documented to host these mites. Very similar life cycle as the tropical rat mites. The protonymphs and the adults feed on blood. They are arenatochus, and they can wander very far in search of a blood meal. The image on the top here shows a window-mounted air conditioner unit. And a lot of times these will have plastic housing as part of the unit. And if birds get in there and establish a nest, that's a source of these mites that's right there next to the structure. And in fact, some openings. If you've ever installed one of these window units, you know that you're not going to get a perfect seal most of the time. So you will get mites moving right into those gaps. Another species that we see from time to time is called the spiny rat mite. And this one also parasitizes rodents, and it's especially common with the Norway or sewer rats. It's a larger mite. It's also heavily sclerotized, so it has a dark body, and it's imminently visible. You can see these guys very clearly. The interesting thing about the spiny rat mite is its chelicerae, those are the fangs, are not capable of piercing skin. Instead, it feeds on secretions from damaged skin. So it's associated with wounds. And so that means if it is wandering around, it's not likely to cause the same kinds of dermatitis that the rat mite and tropical rat mite and fowl mites do. And this one is known to die within about a week without its food source. So this is kind of a secondary pest that we see in structures. Another one, which is very common with poultry production all over the world, is called the poultry red mite, or sometimes called the chicken mite. And you can also see this one. They can actually get quite large when engorged, up to 2 millimeters. And these guys also feed on blood, and they can cause major dermatitis if they get indoors and bite humans. And they can persist for a long time without a host. The ecology of this mite is kind of like a tiny bed bug, where they will live in cracks and crevices near the host, come out, take a blood meal, and then go back to their crack or crevice to digest it. This one is becoming more common, especially because if you look at the image in the lower right here, this is a poultry pen or coop that's right next to a home. And I believe there's even a window behind that coop. And so if you have large populations of these mites developing right next to a home, they will find their way in and they will bite people. I grew up in Florida where everybody talks about chiggers, but in California, I've heard this quite often. They say, oh, we don't have chiggers here. But in fact, there are a few areas of the state where they're exceedingly common, especially seasonally. So usually early summer in the East Bay Hills, also the hills above Ventura, Santa Barbara, you can find chiggers. What you'll notice if you look at the image on the left here, that's a larval mite. It only has 6 legs. And in fact, the trombiculid mites, only the larval stage is a blood feeder. So this article on the right from the New York Times that talks about chiggers actually is showing probably a pseudonymph for an adult, which is not a parasitic form. You know, these are free-living predators most of the time. So the point of this slide is to just let you know that there could be landscape species that are causing dermatitis. Usually with chiggers, you're going to see a pattern of dermatitis near constrictions in clothing that were worn during gardening or hiking. So along the waistband, in the armpits, the cuffs of the sleeve, sock lines. These are areas where chiggers are going to settle and cause dermatitis. And they can be prevented, or at least the dermatitis can be prevented by taking a hot, soapy shower after you've been in an area in early summer where you expect there could have been chiggers. And that dislodges these wandering larval mites and prevents the dermatitis. So what do pest control professionals do out there? Commonly, if you have a good biting mite program, it looks like this. You have to control the primary host. And so here's a Norway rat that's been trapped. Some kind of program needs to be instituted to control or remove the primary host and its nesting material, if possible. We can also try to use exclusion tactics to keep these primary pests from coming back in to the attic or the crawl space, or the eaves if we're talking about birds. Sometimes pest control uses oils or soaps as contact acaricides, and these are effective if they contact the crawling mites, but they can cause damage to the furnishings depending on the formulation. So make sure you read the labels. A lot of pest control operators have told me that they use pyrethroids or pyrethrin insecticides. These will kill wandering mites unless they're resistant, but they need to be labeled for indoor application. And they really should be restricted to areas that are not going to be contacted by humans or pets or other creatures that are indoors. So really, we're talking about crevices, maybe spot treatments of certain surfaces. But the biggest need that I see out there in pest control is better monitoring tools for these biting mites. And so I designed a research project that began in 2021, where we first wanted to create a collection of biting mites that we found here in California to determine what species we had. From that collection, we wanted to take photos and I create some guidelines for identification that could be used by pest control, and then develop some monitoring protocols that we can use in places where we suspect we're dealing with biting mites so that we can move forward with control measures. So this project is funded by the state, California Department of Pesticide Regulation, and it includes a lot of partners. Most importantly, vector control agencies all over the state. So this is my colleague, vector biologist Wade Lee. And so he's an amazing resource here in Alameda County, but there are other vector control agencies that are aware of this issue and are out there to help pest control with identification, especially. So as part of this project, we've already made about 40 collections, mostly from California, but we've also received submissions from pest control operators in other states and regions. And so what did we find? When biting was assumed, when we had dermatitis, tropical rat mite is the most common species that we have recovered. It's about 80% of our biting cases. The fowl mites, northern fowl mite and southern fowl mite, represent about the majority remainder of these biting cases. It's important to note here that the tropical fowl mite is only found in Southern California. I have not received specimens from anywhere else of tropical fowl mite. The northern fowl mite, however, is present everywhere. We've had submissions from the East Coast, from Canada, and it's a very, very common species. And again, it's a primary pest in poultry operations. We also got a lot of submissions of some conspicuous species called concrete mites or sometimes sidewalk mites. These are common in the springtime. And then, of course, we did get some submissions of chiggers. Everybody has seen these, aggregations or at least high density populations of these mites. In my area, it's usually in May. In Southern California, it may be even earlier in the year. These mites are gathering pollen grains. They are omnivorous. Some species parasitize other insects. Some species are predaceous, but most of them will gather pollen in the spring. And so I think what we see is two things here. It's easiest to observe these mites when they're on concrete or another contrasting surface than if they're in the leaves or soil litter. And then, also I think it's easier for them to gather pollen grains that are on these hard surfaces. So these guys are important because they're conspicuous. Your customers will see them. They can crawl around on patio furniture, and they are known to bite people, probably as a defense mechanism. But there are cases of dermatitis caused by bites of this genus, Bolaustium concrete mites. Okay, so let's talk about monitoring. Over the years, we have observed some really interesting behavior, and we have considered some potential tools for trapping mites indoors. First, the literature and observations show us that these mites exhibit what's called negative phototaxis. So they move away from light. You're going to find them in dark areas, crevices, corners, under things. They also exhibit what's called thigmotaxis. So they are attracted to tight spaces where the surfaces are touching their bodies. So again, think of like a tiny little bed bug. They really want to get into a crevice and be protected in that way. The most interesting observation here is we have observed what's called negative geotaxis. This means they move away from the ground. They tend to move up. So when you see them indoors, they often are crawling up walls or along ceilings. You don't often see them on the ground. And we think that this is a host finding behavior because these mites will actually drop, or it seems that they will drop onto victims from above. And so there was a really great article published by Pest Control Technology. I think it was in 2021. Josh Shoemaker was one of the authors, and he says it seems the mites travel up walls and across ceilings where they drop on sleeping victims below. And so we've seen this in lots of cases. If you look at the lower left, this is a heating supply vent above a toilet in a residential bathroom. And so in this case, the people said that they experienced bites when they were sitting on the toilet. And what was happening is there was a roof rat infestation in the attic, and the heating supply ducts ran through the attic. And so the mites were crawling around on the surface of these ducts, and then they were able to exit from the exterior of that heating supply duct, crawl around this faceplate, and drop right onto the people that were sitting on the toilet underneath. Very frequently, we will find biting mites on the center of horizontally placed glue boards. And we think this is because the mites are exhibiting this host finding behavior where they're dropping from above. And I've actually heard from a number of residents dealing with these mites that they first saw the mites when they were in their bed on their phone, scrolling through whatever social media black hole they were in, and they saw these little crawling specks that fell on their phone. So we don't know if that means the mites were attracted to the heat or the light of the phone, or really if it was just incidental and there were mites falling all over the place, but only being observed on the illuminated screen of the phone. So this is an exceedingly common phenomenon. Now, of course, the other hypothesis for why mites get trapped in the middle of glue traps is static electricity. So this video here shows some of those concrete mites within a Petri plate with a cover on it. And there's some static electricity generated within this plastic dish. And you can see how it just vaults the mite all over the place. So this may be happening in some cases, but we think that the majority of the cases where you see mites in the middle of a glue board is because the mite fell from above or dropped from above. We've done a couple of lab trials trying to look at different stimuli as attractants for these mites. First, we looked at heat. So I set up my experiment this way. We had some live tropical rat mites that did not look like they had fed, so they were hungry. We collected them from a residence in Oakland. We released them into a lab tray, which is, about half a meter square on each side, more or less, and used petroleum jelly and tape to make sure that the mites could not crawl out of this environment. And then we introduced several different monitoring devices, glue boards, passive bed bug monitors that basically are Harbridge traps, and then our own Harbridge trap design, which is just a 5 centimeter square of corrugated cardboard. And on one side, this image doesn't show it, but on one side of the arena, we introduced a hand warmer. And these are the same kind of hand warmers that you use when you're skiing. you break the package or bend the package, and it starts a chemical reaction that produces heat over a 10-hour period. So in this case, we had 2 different hand warmers that were used in series, so that we had about 20 hours of heat release. And 24 hours later, we counted where these mites were found. Yeah, here's the methods that I just described here. Mites were counted and then assigned to the substrate. Where did we find the mites 24 hours later? Were they on the side with the heat or the side without the heat? And so the results here, most of the mites remained in the release tube. They didn't go anywhere. 10 of them were found wandering around on the tray without interacting with any of our monitoring devices. And so really only a handful were trapped within our monitoring devices. You can see that these guys are visible. If you look at the video on the left, you can see 2 dots there. One of them's moving a little bit. Those are the mites. We concluded with these quick and dirty lab trials that heat by itself does not appear to be effective as an attractant. So next we started looking at carbon dioxide. We know from the literature that carbon dioxide is extremely attractive to blood-feeding arthropods. But indoors, carbon dioxide is difficult and potentially dangerous to deploy. So we opted for use of a sugar yeast reaction. And we got the recipe from this bedbug paper published in 2015, where basically you're mixing yeast and sugar with warm water, and you're expecting emission of carbon dioxide over the period of about 10 hours. We also wanted to look at different orientations of trap deployment. So we use these traps in traditional horizontal placement, but we also applied some vertically on walls. And we also looked at heat. So we really had 3 different factors being considered here: addition of carbon dioxide, addition of heat, and then vertical versus horizontal placement. And so what we found, and this was at a public school in San Francisco where we were having bites, and we had already recovered some mites. We trapped over a 72-hour period. It was over the weekend, when we could deploy these traps without them being tampered with. And we did recover mites in a short period of time in that room where bites were being reported. All of our mites were caught on traps that included stimuli. So that means they either had heat or carbon dioxide or both. And so when we placed these traps horizontally, we caught mites in the center because they were dropping down onto the traps. When we placed these monitors on walls, we caught mites on the edges, suggesting that they crawled into the glue surface. If you want to watch some really cool videos that actually they just won an award, video production award, you can find these on YouTube in both English and Spanish. There's kind of a 3-part series that talks about what we did and why we did it and what we found. So what do you do if you catch some mites on glue boards like this? Well, if you want to identify them, you have to prepare them for slide mounting. So unfortunately, if you want to identify the species, the only way we have found that you will be successful is if you mount these guys on a slide and look at them under a compound microscope. And this requires first lifting them from the glue board, or if you're using double-sided tape, lifting them from the tape with mineral oil, transferring to ethanol, clearing their bodies to remove their blood and their gut contents. And there's different clearing agents that can be used. If you don't have access to lab chemicals like Nesbit's or lactophenol solution, you can actually use mineral oil or in a pinch even deionized water. But the trick here is heat. You want to have some gentle heat, about 50 to 55 degrees Celsius for 12 to 24 hours to clear the contents of these guys so that you can see the characters that you will need to identify them. Then, of course, you transfer this specimen to a microscope slide. Now, when we first began this project, we wanted to develop guidelines based on macro images that we could take with a dissecting scope. And so that's what you see on the left here. And this is actually a stereo microscope photo using image stacking software. So it takes images at different focal points and then smushes them all together. So it looks like more of a 3D image. But try as we might, we cannot identify species looking at these macro photos. You cannot see the characters that you need to see to get to the species level. So in fact, you do need to clear and slide mount these specimens. And I'll show you some of the characters that you're looking for. If you want to rule out those landscape species like the chiggers and the concrete mites, you want to see whether or not this is a mesostigmatid. And so mesostigmatid mites can be identified by a pair of stigmata. These are respiratory organs on the lateral edges of the body. And then also, what's usually easier to see is a structure called the apitel. It's like a little claw on the anterior margin of the palpal tarsus. And so, of course, these are tiny little structures, and you can see why you have to slide mount these guys. And this is just to get to the right order. And of course, if you don't see those structures, you might consider that you're dealing with an outgroup of one of these landscape species that cannot be managed in the way that the indoor species can. Chiggers, Balaustium, or what we call itch mites, which can be associated with straw or hay, or sometimes deciduous trees like oaks. Other discriminating characters that will help you get to family level, the presence of the tritosternum, which is a soft tissue, kind of a deedly bopper that hangs off of the ventral surface of some of these mites. You can look at the number of hairs on the genitroventral plate. And so these characters are used to rule out some of the outlier families within mesostigmatids. You can also look for these structures called corniculi. They're kind of some sclerotized sheaths or covers on the chelicerae that are associated with the lelapids, which is where we find the spiny rat mite, which, remember, is a secondary pest, or at least of secondary importance compared to our main species of interest, which are in the Ornithonissus or Macronicidae. And so you can look, hopefully, if you find the chelicerae, these are the fang-like structures that come out of the mouth of these mites. If you're lucky, sometimes when you mount them, you can't see these, so you have to find a good specimen. But if you look at the shape of these, if they're shaped like a spear or a whip, that generally indicates the family Dermanicidae. That's where we find the chicken mites, also known as the poultry red mites. There's also a species called the house mouse mite that's in this family. The lelapids, this is where we have our spiny rat mite, or the macronicids. So the lelapids have these little lobster claw chelicerae, and the macronicids have what we call shear-like chelicerae. It looks like little scissors without claws. They're just kind of straight shears. And then, of course, to go even deeper, there are characters that we can use to identify the species. So in this case, we're looking at the number of setae on the sternal plate to identify the tropical rat mite and distinguish that from other species within Ornithonissus. So you can see it gets complex quickly, but if you have a lab, if it's equipped with a compound microscope, you can achieve this level of identification in-house. We can also look to our partners like vector control or sometimes ag department in your county to help with these kinds of identifications. So let's talk about control. There are a lot of really effective non-chemical things we can do. The most important, remove the primary host. You have rat mites because you have rats. So let's control the rats, or else these mites will continue to reproduce. So if you want to stop reproduction, you have to remove the rats or the birds, or move the chicken coop further away from the house, that kind of thing. You can try to seal the access points between where you think the mites are reproducing and where people are getting bitten, but this is much harder to do than it sounds because these guys are smaller than a millimeter. So you're going to have to eliminate all cracks, all crevices, all utility penetrations, all gaps around windows and doors in order to seal these access points. Vacuum is very effective at removing wandering mites. As is surface cleaning, I should mention. So, surface sanitizers or cleansers that are used indoors. Laundering will kill all life stages, just like with bed bugs. And then this is something that we've considered doing, but if you think you know where mites are coming in, you could use, in some cases, double-sided tape as a barrier to prevent them from crawling to where people are. This is difficult because they can drop from above. So if the source is above and they're dropping from above, it may be very difficult to exclude access to the humans below. We can also consider chemicals, but these are really only targeting the wandering mites. And so I have heard from pest control colleagues that they have seen good results with essential oil products. These are contact acaricides. They will kill, but you have to actually contact the wandering mites. And then, of course, since some of these are oily, they can damage wood or other surfaces, veneers and whatnot. A lot of people want to reach for the residual insecticides or acaricides, and really we're talking about pyrethrins and pyrethroids. These are nerve toxins. Some of these may also damage furnishings, so you have to make sure that they are labeled for use indoors. And if used correctly, they may, in theory, offer 2 or 3 weeks of residual control. So if a mite walks over that treated surface, it will die. However, we see a lot of resistance to pyrethroids out in the environment within these mites. And of course, anytime you spray a surface indoors, you are creating potential for human exposure, especially to children or pets that are on the floor crawling around if you're making applications to the floor. What's really interesting is that in veterinary pest management, there's been good success with insect growth regulators, specifically hydroprene and methoprene. But this is only used against the reproducing population. When you have wandering mites biting people, it's usually adults. And so those adult mites will not be affected by IGRs. So I just wanted to show you this paper. This came out from a colleague, Professor Amy Murillo down at UC Riverside, 2017. And they were looking at northern fowl mites in poultry production and their susceptibility to different acaricides. And they were not able to calculate the LC50, the lethal concentration required to kill 50% of the population for permethrin and deltamethrin, which are pyrethroids, because no mortality was observed, even at 1,000 parts per million. So this suggests that there are high levels of pyrethroid resistance out there. Hey, does this sound familiar? German cockroaches, bed bugs, we've got pyrethroid resistance out there. It's very, very common. So we cannot rely on these kinds of residual insecticides. If you want to learn more about this pest system, we did publish one article in the Green Bulletin back in 2020, and it was turned into a blog post, and that's just called Detecting and Controlling Biting Mites Within Structures. It covers a lot of what I've covered here today. And there's a new guide that's out that was published by Ben Huddle. He works with Rollins, Rollins Pest Control. the parent company of, I guess it's still parent company of Orkin. And I was included as a co-author, as well as a really talented acarologist, Dr. Pavel Kimlov. And in this guide, we show pest control operators ways to hopefully identify the mites that they are observing at their client sites. So this is freely available within a newly published NPMA guideline. So take a look at that, and thanks to Ben for developing this guide. That's all I've got for you guys. Hopefully we have time for a few questions. Thank you very much.